Now move on to the slide labeled Lung 3 of a squamous cell carcinoma. By eye, observe that the tissue section has a semicircular shape. Black ink is present on one of the edges to indicate the margin of the resection. The outer region consists of collapsed alveolar tissue, while the inner darker staining core contains a mixture of tumor cells and normal connective tissue. No cartilage is apparent, suggesting that the tissue was taken from a more peripheral region of the lungs. In addition, there are small nodules of lymphoid cells scattered throughout the tumor mass. Under low magnification, examine the edge of this section and observe the ink indicating the margin and the normal alveolar tissue immediately adjacent to it. There is some edema or swelling of the alveolar tissue due to the presence of the tumor mass, which can be seen on the left side here. Next, examine the edge of the tumor mass under low power. The squamous cell carcinoma is found in small convoluted clusters that are separated from each other by the darker staining normal tissue that has been compressed. Switch to higher power and scan the tumor mass. In many places, you can observe collections of lymphocytes adjacent to the carcinoma cells, as shown here. In other locations, you can observe that the tumor cells consist of nests of cells with a distinct basement membrane. The tumor cells are large and have distinct cell-to-cell boundaries. Some of the larger nodules have necrotic centers. There is a degree of pleomorphism with regard to both cell size and nuclear size. However, keratin pearls and desmosomal junctions are not obvious, suggesting that this sample is only moderately differentiated. Now examine the slide labeled Lung 4 that contains both large cell carcinoma and adenocarcinoma. This slide is notable for several reasons. First of all, the diagnosis of this specimen is somewhat ambiguous. It contains two different types of tumors, which can occur occasionally. The large tumor mass appears to be a clear cell variant of large cell carcinoma. In addition, there are several smaller regions that appear to be adenocarcinomas. And secondly, this slide is particularly well preserved, and for this reason, you are directed to identify a number of different normal tissues. The most prominent feature that you can observe without magnification is a dark basophilic, that is blue, line on one side and several smaller spots on the other side. This is cartilage that is associated with a bronchus, indicating that this fragment was taken from the hilar region of the lungs. Please note the locations of the adipose tissue, the nerve tissue, the adenocarcinoma, and the large cell carcinoma. Under low magnification, examine the region of the large piece of cartilage. Identify the normal structures that are found in the wall of the bronchus, including the cartilage itself, the respiratory epithelium, and the mixed serous mucus gland. Switch to higher power and examine the structures in greater detail. Locate the glands that are between the cartilage and the bronchial epithelium. They contain both mucus units that are light staining and serous units that are darker staining. Now move the field of view to the lower right-hand side of the cartilage, assuming that the slide is in the same orientation as shown before. Locate the nerve tissue that is within the adipose tissue. Note that it is encapsulated and has a wavy appearance. In some places, you can see axons passing through a myelin sheath. The myelin sheath itself has a crackly appearance. Examine the normal alveolar tissue on the slide. Identify the large gray cells in the air spaces. These are macrophages, also known as dust cells. Some may contain the remnants of blood that had been phagocytosed, now termed hemosiderin. Switch back to low magnification and scan the large tumor mass and note the following. First, the border between the tumor and the normal tissue is indistinct due to a heavy infiltration of lymphocytes. Clearly, the tumor is stimulating an immune response. And secondly, while the tumor mass is large, there is only a thin rim of actively growing cells around the edge, and this is mixed with the lymphocytes. Now examine the tumor at higher magnification and observe that most of the tumor mass is composed of large vacuolated cells and other regions of frank necrosis, that is, dead cells and debris. The large vacuolated cells are characteristic of the clear cell variant type of large cell carcinoma. Now scan the region around the smaller fragments of cartilage and locate the gland-like tissue that is probably adenocarcinomas. They form acini that are lined by a single layer of cells, and their lumens may contain mucin. Also note that they are surrounded by heavy infiltration of lymphocytes. The final slide in this laboratory is labeled Lung 5 and contains an example of a large cell carcinoma. Remember that large cell carcinoma is a diagnosis of exclusion. That is, it is so poorly differentiated that it cannot be distinguished as either squamous cell or adenocarcinoma. Examine the slide by eye and observe that it consists of a single semicircular shaped section with two basophilic staining tumor masses, both of which are located at the edge of the specimen. Also observe a lighter staining region next to the smaller tumor mass. This is normal alveolar tissue that has become compressed. Also, on one edge, there is some black ink to indicate the margin. In addition, there appears to be a small region of the serosa that lines the outside of the lungs. I want you to examine this region first. Under low magnification, look at the edge of the tumor and locate the black ink that was used to mark the margin of the sample. Switch to higher power and find the serosa connective tissue that is located on the outside of the lungs. Note that the carcinoma appears to be invading through the normal alveolar tissue in this region, but has not quite reached the serosa. Now move the slide to the border region of the tumor mass. The normal alveolar tissue in the surrounding area is quite thickened and filled with debris. This represents interstitial edema, which is probably a secondary consequence of the tumor mass that has caused an obstruction in this area. Examine this normal alveolar tissue under higher power and observe the large round gray staining cells in the airway. These are macrophages, also known as dust cells, that have phagocytosed debris from the air. Next, examine one of the tumor masses under low power. Note that it consists of a cluster of tumor cells, and the clusters are separated from each other by a thin layer of collagen. Some of the tumor masses have necrotic centers. In the center of this figure, you can see that the tumor cells have invaded the lumen of a blood vessel. Now switch to higher power and examine the tumor cells near the edge of the tumor mass and next to the wall of the lung. The tumor cells are in small clusters invading the surrounding connective tissue. They have a polygonal shape, which is characteristic of large cell carcinoma. This now concludes the laboratory on bronchiogenic carcinomas.